Andersen. I'm an associate professor at the Department of Neurobiology Research here at SDU, but I'm also a research associate professor at the Department of Neurology at OUH, and finally I'm also the vice head of BRIDGE, the brain center here in the region of Southern Denmark. For me it's a pleasure to be able to introduce Jeppe Vinther. He's not only a friend, he's also a great scientist. And he's going to present some of his data today. And just a very brief CV on Jeppe. So Jeppe graduated from the University of Copenhagen in 2000. Then he did his PhD at the Faculty of Health Science. And now he runs his own lab, also at the University of Copenhagen. Jeppe has been granted several huge grants to support his research. And I'm looking forward to hearing you talk. Welcome.
Thank you. Yes, so I'd like to thank for the invitation. It's great to be able to present some of our data here. I made back and forth with Charlotte about the title, and I think the first one I sent was even more technical than this one, but then I tried to make it more interesting, and this is the title that we ended up with. So I'll talk about two studies that we've been doing on RNase H, which is an enzyme that cleaves RNA when in duplex with DNA. So here we have this heteroduplex, and this enzyme is able to specifically recognize the heteroduplex and then cleave the RNA strand specifically. So I'll talk about this enzyme today. In my lab we also do some structure probing of RNA, and also look at RNA modifications, but I will not discuss these things today. So I'll give a brief introduction to RNase H enzymes. What are they? What are they doing? And then talk about this class of drugs called RNase H-recruiting oligonucleotides, also called gapmers among friends. And then we have these two studies that we've been doing, looking at the specificity of these gapmers and also on preferences of RNase H enzymes. Yes. So as I mentioned, we have this class of enzymes, RNase H enzymes, which are basically present in all living creatures and also in many viruses. So it's an ancient enzyme, and it's quite important. It's essential for development, and also a virus like HIV cannot work without its RNase H activity. So in humans we have two different RNase H enzymes. We have RNase H2, which is a trimer. Mostly works in the nucleus, and it's able to cleave out a single RNA embedded in a DNA duplex. Then there's RNase H1, which is more closely related to the bacterial RNase H enzymes. It's a monomer, and it requires a stretch of at least 4 RNA positions in a double-stranded duplex to cleave the RNA. So as I said, RNase H1 is quite highly conserved between coli and bacteria, or between humans and bacteria, and even more so at the structural level. You can see that these proteins are virtually the same structure, even though there's quite a lot of sequence divergence. So human RNase H1 is localized both in the nuclear plasm and also the cytoplasm, and mostly in the mitochondria, where it has its main function. It's not completely worked out, but it's essential for replication in mitochondria to work. So for this talk then, it's important that this class of drugs, the RNase H-recruiting oligonucleotides, is completely dependent on the activity of RNase H1 for activity. So this has been shown in knockout mice. So turning to these oligonucleotides, then it's the basic principle is quite simple. You have a DNA oligo which is complementary to an RNA sequence, so you can have hybridization. This heteroduplex will be recognized by RNase H, which will cleave and then leave the ends of the RNA unprotected, and then it's believed that the body of the RNA is then degraded by isonucleases. This is quite efficient. These oligos, they can get into cells and they can degrade RNAs both in the nucleus and in the cytoplasm. So this means that introns, for instance, if you have an mRNA with introns in it, then these oligos will also inhibit the pre-mRNA by cleaving the introns before they're spliced out. So this is an advantage of these oligos if, for instance, you compare with siRNAs, which many of you may know. They work in the cytoplasm and will not target pre-mRNAs. Yes. So in order to make this into a real a drug-like molecule, you have to modify the oligos. So if you just give a DNA oligos, a primer, like the ones we use in PCR, then it will be cleared from circulation very rapidly, within minutes. So in order to keep these oligos in the circulation, then they are modified, and it's typically with this phosphorothioate modification, which is changing one of these oxygen atoms to a sulfur. This changes the charge on this position, making this a better binder of proteins. So in this way, it will bind to the abundant plasma proteins, and in this way stay in circulation and not being filtrated out in the kidneys. It also stabilizes the oligo as such, so it's not degraded as rapidly. So here's a study that was done by some of our collaborators at the Danish firm that used to be Centaurus, but now is Roche. So here they have given this, I don't know if you can see what it is, but this is actually a really, really thin slice of a mice. So they've given the oligo, which was radioactively labeled, to the mouse, waited for some time period that I cannot remember, and then they froze the mouse in liquid nitrogen and cut it into a very thin slice, and then they exposed to film. And here, the darker the signal, the more oligo was in the tissue. As you can see, it's very much of these oligos end up in the liver. Also, quite a lot in the kidneys, and then there are less in the other tissues. And I guess this is kind of an image of the perfusion of these different tissues. So you know that liver and kidney are highly perfused. So this is also shown here. So this means that most of the drugs that people have tried to develop are liver targets, or targeted against liver molecules, or genes expressed. Yes. But also, in order to make these oligos even more efficient, then there are other modifications. And one very used modification is the log nucleic acid. And here in Odense, I guess many of you know this modification. It's invented by Jeppe Vinther and has been a big improvement for these oligos. So this is the ribose of a DNA molecule here. So we have the base sticking up here, and we have the ribose unit, and this is then the backbone here. Normally DNA is in this C2 endo conformation. RNA likes this C3 endo conformation, and this RNA binds with higher affinity than DNA, and for these oligos we would like the affinity to be higher. So this RNA modification makes a bridge here between the 2' and the 4' position, and this way locks efficiently the ribose in the C3 endo conformation, which increases the affinity of the oligos. Also, it makes the oligos more stable. So that way, it's a really neat modification for these oligos. So Jeppe took out a patent some years ago, and this patent has been the basis for Danish companies, Centaris and Exicon. Centaris is now bought by Roche, and Exicon is bought by QIAGEN, and the patent is running out. So soon, I guess many more people also in normal basic science will be using the LNA modification, because in a short time they will be cheap to buy. But for these oligos, they are very efficient, and especially Roche has been using this modification for their drugs. Then I'll just mention that these oligos can also be made more efficient by conjugating different ligands to the end of them. Typically these ligands bind to some specific receptor on the target cell. And the most efficient molecule that has been discovered is this one called GalNAc. Binds to a highly expressed receptor on the surface of liver cells. And this increases the potency around tenfold of these oligos if you conjugate this molecule to the oligo. Many of these companies are trying to find other conjugations for the oligos which could target it to other tissues. And there's been a recent success that it seems that they found a ligand that can target the oligos to beta cells, which could be very interesting. So these oligos, in general called RNase H-recruiting oligos, also sometimes gapmers, and it's because they are designed like this. So this will be a gapmer. We have LNase here in the ends, the flanks of the oligo, and then we have phosphorothioate at the backbone here, not really shown on this. The LNA in the ends increases the specificity of these oligos, the affinity, and then keeping the DNA in the central part of the oligo makes the oligo able to recruit RNase H and cleave this target RNA molecule. So this is a gapmer. There's one drug approved in this class, and there are many under development. But even though people have been working on this for many, many years, there are not that many successes. Hopefully more will come in the future. Yes. So now we're getting close to where our research come in. Antisense oligos are really neat in that you can target it against any RNA molecule. So it could be a non-coding RNA, it could be an intron of a pre-mRNA. So this kind of makes the entire genome druggable. So there are big upsides of this technology if you could make it more general and make the platform more efficient. So when a company like Roche, they find some target that they want to make a drug against, they have the sequence of the gene, so they know the exact sequence of the mRNA. So they know we need to target this RNA sequence. So then, in principle, it's very easy to design the drug. You take the reverse complement of the sequence, and then you have your drug. But of course, you want to have the most efficient of these many possibilities. And because you can target introns as well, then you have many, many millions of possibilities for which sequence to target. So of course they try to, before designing and testing, they try to use computers to come up with the most, the best candidates for efficient molecules. Yes. And of course, you would like to somehow optimize the hybridization between the oligo and the target. Also, it may not be a good idea to target a region that is folded in a secondary structure in advance, so they also look into this kind of thing. And of course, you want to avoid that the oligo has complementarity to other places in the transcriptome. That would give you a risk of having off-targets. And then, of course, you would like to recruit RNase H as efficient as possible. So, in this first study that I'll talk about, then we looked a little bit on this, what defines an RNase H off-target. And then in the second, we tried to look into what is the sequence preferences of RNase H1. Yes. So this is kind of the starting point. We would like somehow from the sequence to come up with the best possible guesses on active gapmers. And by knowing more about these things, then the hope is that we can come up with better suggestions. So typically the company, when doing this kind of thing, they predict 200 different oligos, and then they screen these in vitro before going on with 1 or 2 molecules. And of course, these 200 that they screen in the first, they should be of the highest quality possible. And that's the idea of this, to make these molecules as good as possible. Yes. So now we come to the results. The first thing we did was to make what we call a library sequencing. The idea is to try to figure out what sequences can be off-targets for these gapmers. So what we have here is a plasmid. It has a luciferase reporter in it, so it's driven by a promoter here. And then in this small part of the 3' UTR, we put in a random sequence. So we have, in this specific spot, we have all different possible sequences. This sums up to around 16,000. So in fact, we have a pool of plasmids, 16,000 different plasmids, just differing in this exact position of the plasmid. Then we transfect it into cell. The mRNA will be expressed from the plasmid, so we'll also carry this sequence here. So in principle will be 16,000 different mRNAs expressed in the cells. Then we isolate RNA, do reverse transcription, and build sequencing libraries. Then we sequence, and then we can compare the counts we get when sequencing the plasmid with the counts we get when we sequence the cDNA. So if all of these mRNAs were expressed with the same efficiency in the cell, then we would expect to have the same relative count between the cDNA and the plasmids. If an mRNA was downregulated, perhaps because of this sequence here, then we would expect to have fewer count for that specific short sequence. We can also do some kind of co-transfection here, and in this study we co-transfected with this gapmer that is has the potential to bind in this specific location. If we do this co-transfection experiment, then we could make a study where we transfect in a control and then the gapmer, and then we could compare the cDNA counts, and this would tell us did the co-transfected gapmer have an effect on all these different sequences. Yes, so please interrupt if you have questions or I move too quickly on some of these things. So we started by looking into the if we could replicate in this system, and in 2 different PCRs on the plasmid we have very high correlation. So each point here is a count for a sequence. So there are 16,000 points here, and we count in the two experiments how many times do we see the different 7-mer sequences. So this looks quite nice. Then we tried to do the transfection and sequence on the cDNA and compare with the counts we got from the plasmid. And here we also have fairly decent correlation, but less than what we did from PCR in the plasmids 2 times. And of course we hope that the differences we see here are due to regulation in the cell, so that it is the small difference in the 3' UTR that makes these counts different. So we started by looking at microRNAs. MicroRNA regulation depends on binding of the microRNA to these microRNA sequences. And in principle, in our library, we have all possible microRNA seed target sequences. So, and this was actually, to be honest, the reason that I started this project in the first place. It's because I thought that we could have a way of profiling the efficiency of microRNA regulation in cells. So we do have a signal here is showing the different curves are for the 7-mer sequences that are complementary to microRNA seeds. And here is for the microRNAs we know to be expressed in the cell that we're using, and then all the other motifs. And then here we are having the relative ratio between counts in the cDNA and the plasmid, and we can see that the curve here is shifted, showing that the words or 7-mers that are complementary to the microRNAs, they are downregulated compared to all the other words. So that was a nice validation. So we can show this when we look at all the microRNAs as a group, but unfortunately we could not, our signal was not good enough to see the individual microRNA, whether they were regulated. So I thought what to do with this, and then we thought, okay, let's try to see if we can look into gapping this with this system. So instead we then take our plasmid pool and co-transfect it with the gap. And we started out using 2 different gapmers here. So we have the flanking sequence here match the sequence in the plasmid, and then we have a specific sequence here in the middle. We then got our data back, and here every point is one of these 7-mer sequences. We're showing the ratio between the control transfected and the gapmer transfected, and then on showing the abundance of the different words in the library. So the ones that are really highly expressed, we will get better data, and the ones that are really lowly expressed, you can see we have high variation because we have simply too few counts to determine the relative number in a statistically significant manner. Then each of the points, we calculate the binding score, that is, how many base pairs do we observe between the sequence and the oligo we put in to the system. And here, it was a quite nice day at work when we saw that the sequence that were complementary to the oligo we put in was indeed the sequence that was most downregulated. So that was quite nice, and we can also see that the ones that are somewhat downregulated, they have one mismatch compared to the sequence. The same for another oligo here. Again, the 7-mer that was most downregulated was the one that was the complementary to the oligonucleotide system. Very nice. Then we could also see that here this one seems to be more specific actually than this one. Here we have more of these binding score 11 and 12 that are downregulated. So these would be off targets for this particular gap. So that was quite nice, and here I think actually already here we can say that it seems that some oligos do have more off-targeting potential than others, as visualized by these plots. Yes. Then we did a control where I should have mentioned this, the bold font here is LNA. So again, these are gapless. We have LNA in the flanks here, and then this is DNA. So we then moved one of these LNA into the middle of the oligo, and this should inhibit the efficiency of the oligo because RNase H cannot recognize long enough RNA-DNA duplex. But this did not come out as we expected. We expected this point to then move into the cloud here and not be downregulated. So this made it a bit, this was our negative control, so we were pretty concerned that this showed that our experiments were not working the way that we hoped for. So we made some new controls here where we again moved the LNA further into the center of the oligo, reducing the size of the gap here to 3 and here to 1. And we can see even for the one where we have the gap of 3, we do have some efficiency here, but when we disperse the LNAs throughout the oligos like this, then the point moves in, and this, I think, showed that our experiments were trustworthy. So that was nice. We were quite puzzled by this. This was a big surprise, not at least for the people at Santaris. They expected this to be a good negative control. Going back in literature, there were some indications that you only need this for base pair DNA RNA duplex in order to have efficiency. So in retrospect, it was perhaps not that surprising. But then we looked at this figure and we compared it to the one we had for the mother oligo here, the B oligo. And it was quite striking that here we have we seem to have a lot of potential off targets, whereas here the experiment looks much more clean. We only have this one point that stands out and seems to be efficiently downregulated. So we were kind of puzzled about this, so we decided to look more into these points that were different in the two experiments, hoping to find out more about this off-targeting. So, and in principle, these off-target could be two different kinds. So we could, here's the perfect match, which is the B oligo. Here we have perfect complementarity. Then of course we could have mismatches. So this would give us a binding score of 12, but we could also have these bulges here where the target is bulging out. So for each of these points we looked into whether it was a mismatch, it was a bulge, and tried to annotate it. And then actually there was kind of a pattern coming out that for the mismatches, that the ones that had largest repression with B, here the mismatches were in the 5' end of the oligo, you could say. And also for the bulges, there seems to be a trend here that, again, the ones that had most repression in the native oligo seem to be localized towards the 3' end of this. So we try to rationalize this, thinking about what is known about the interaction between the duplex and the RNase H enzyme. And I have to say this is quite speculative, so consider yourself warned. The gray stuff here is known interactions between RNase H and the duplex. So you can imagine we have the duplex, so one side of the duplex is facing towards the enzyme. So this part of the DNA strand will be facing the enzyme, and this part of the RNA strand will be facing the enzyme. Perhaps I should use the pointer. The active site here is, of course, on the RNA, and this is the exact position that is cleaved. Then there are two things about the RNase H enzyme which makes it specific for these heteroduplexes. There's a docking of a phosphate group into a pocket here in the enzyme, and the only way that this phosphate can dock in to this pocket is if it is in DNA-like conformation. And then there's a kind of steric, the DNA strand kind of runs into a groove from the enzyme, and there's a steric block that will make it difficult to accommodate the 2' hydroxyl group. So these are the two things that makes the enzyme able to detect the heteroduplex rather than, for instance, double-stranded RNA. So here I kind of put in this LNA into the middle, as we had in one of these molecules. For the other molecules, it would be out here in the frame. So the RNase H can kind of slide back and forth on the duplex. So you can imagine for the one where the LNA is out here, then the duplex could kind of slide in the enzyme, or it could bind in different, probably does not slide, but it could bind in different ways and be cleaved. Whereas when we put in this LNA in the middle of the gap here, then the only place where the enzyme can bind to have a productive cleavage is the way I kind of draw it here. So the only place where we'll have these DNA-like properties will be in this exact positioning on the RNaseH on the duplex. And we believe this is the reason for these differences that we see, that for the original oligo, then the RNase H is able to bind in different ways, so it can kind of accommodate if it's a bulge or a mismatch. But for this other one, it can only bind in one way and cleave, and this way we kind of reduce the off-targeting potential of the oligo. So it's kind of the end of this story. So Rush looked more into this, trying to see if this could be a way forward to make these oligos more specific. So they, I think they checked 10 new ones to see if this worked out. And they actually did seem to be more specific, but they also had lower efficiency. So in the end, they are not really using this strategy. And I guess it kind of makes sense that when you are able to bind in different ways with the RNase H, you have higher efficiency, but perhaps also less specificity. Yes. So just to conclude on this first part, using this sequencing-based strategy, we can detect signal for microRNAs, and we can also detect these transfected, the effect of the co-transfected gametes. I think the results show that RNase H can accept some mismatches and bulges and still cleave targets. And of course, you have to take this into consideration when you're designing these oligos. And if you shorten the gap in the gapmer, then it seems that they can have increased specificity, but unfortunately also in most cases it seems to be less efficient. Yes. I guess I have to speed up a bit to get through this. So I'll move on to the second story here. Again, we are working with RNase H to try to tease out the sequence preferences of this enzyme. And we're using the sequencing-based strategy. In this case, we are not transfecting in our library. In this case, we are making the library in vitro and adding recombinant RNase H to the library. So we make the library in this way. We order an oligo that has these inserts here containing a central RNA gap, and in some cases also 2'-O-methyl. Then we prime and reverse transcribe on this to make it into a duplex that has a central RNA-DNA duplex in it. We then add RNase H and cleave some of it with the enzyme, and of course keep some as control. We do reverse transcription a second time, and then we sequence and count how many times do we see the different sequences. And again, we then look at the relative count between the control and the treated. And this gives us, if we have some sequence here that is very efficiently cleaved by RNase H, we would expect to be reduced more than one that is less efficiently cleaved. So this is just showing the gel. We have the duplex here, and then we treat the RNase, and you can see that we are only cleaving a small part of the duplex. Hopefully this will be the most efficiently cleaved sequences that are cleaved initially. We purify these PCR, reverse transcribe PCR, and then sequence. Again, we look at the reproducibility of this, and it's quite nice. We can see that the controls are highly correlated, and so are the treated. And then when we compare the treated with the control, we have a bit more variability, and this is exactly what we hope. Again, we hope that this difference reflects the true preferences of the enzyme. We look at all these sequences, and for each position, we see if the individual bases are overrepresented or underrepresented in the relative comparisons between these two data sets. And this gives us the preference of the enzyme. So here we did it with the coli and the human one. And this is the sequence preferences that we get out. These are quite similar here, and this is, I guess, what we would expect because they are so structurally alike, these two molecules. We also had this different duplex, and this gives us very similar motive sequence preference. In this case, the signal seems to be a bit better, most likely because here we reduced the gap of RNA to 4 positions, kind of forcing, again, RNase H to cleave in that specific position. Yes. We were a bit concerned because these looked so similar. Was it just some degradation of the duplexes was it really the preferences? So we bought the HIV enzyme as well and used that. So HIV has a reverse transcriptase that has both polymerase domain and RNase domain. And we redid the analysis with this RNase H, and it has a really different preference, which was pretty nice and tells us that this is most likely the true preference of the enzyme. We then wanted to validate the preferences that we discovered, so we took out the best possible one sequence that we predict to be neutral, and then one that we predict to be not cleaved very, very well. Compared to a reference substrate, indeed the substrate that was preferred were cleaved more efficiently. The sequence that were predicted to be avoided was cleaved less efficiently, and the neutral was more or less the same as the reference. So that was quite nice and validating what we saw. But we also puzzled that even though compared to the reference we observed what we wanted to see, that we could validate, but more material was cleaved when we used this avoided sequence. So we went back and thought about this. Of course, here we have the enzyme that binds the DNA RNA. We have a duplex which can then be cleaved. The RNA strand is cleaved and then it's released from the enzyme. And basically, because we are doing this PCR, the only thing that we can observe in our experiment is these first parts of the cleavage, but not this last release. So we thought that our results here could be explained if the preferred sequence was also staying longer with the enzyme and this way inhibiting further processing. So we looked into this, we made this kind of product mimic that could not be cleaved, and indeed the preferred sequence here inhibited the cleavage of the reference, so suggesting that the preferred sequence binds with high affinity to the enzyme and stays there, so that it cannot cleave the reference. We also did an experiment where we used a high amount of RNase H, and in this case the preferred sequence here is cleaved more efficiently than the avoided sequence, as we would expect. Yes, so I think I'll skip through this a bit fast. So we used all these data points that we have to make a model that could predict cleavage from our data. And we used both a single nucleotide and a dinucleotide model for this. The dinucleotide model was more efficient than the single nucleotide, and if we used even longer words for the model, then it did not improve much, so we went and used this dinucleotide-based models for prediction. We looked at the crystal structure of RNase H1, and this is a duplex that is crystallized with the enzyme. And in principle, this enzyme could have bound in any location of the duplex, but it bound in this particular region. And when we use our model to calculate the scores, then we see that this is indeed the position on this duplex that we predict to be the most preferred. So that's kind of validating our model. We also looked at some data for many grapnels and their efficiency in cell culture to downregulate target genes. And yeah, it's a bit busy figure, but we are correlating the score we get out of the model with the inhibition observed in the experiments, and see how well it correlates. And we can see as long as we are within the cleavages within this gap, which then we have fairly nice correlation, but if we outside, then there's little or no correlation. So this kind of shows that there are some correlation between what our model predicts and what was observed for these oligos in cell culture. So we looked a bit more into the HIV RNase H as well, and you can see that it seems to have more distinct sequence preferences than the human RNase H. So we looked more into this. We had to do some tricks to build the model here, and I won't go into details with this, but we were able to build a model that can predict cleavage by the HIV RNase H. So here, as I said, we have again the duplex, we have the RNase H domain, and we have the polymerase domain here. The active site of RNase H is here. So when this enzyme is working in real life, we have polymerization going in this direction, and then sometimes the RNA strand is cleaved by the RNase H. It turns out that the duplex has to bend in order to fit into both active sites here, and we are kind of suspecting that the signal that we see here is a signal for the sequences that are able to bend so that you can have simultaneous polymerization and cleavage by RNase H. So we looked at the HIV genome sequence and predicted cleavage across the sequence. So the more going down here is more predicted cleavage. The replication of HIV is pretty complex. It starts by using a tRNA, cellular tRNA as a primer, which is then extended by the polymerase, cleaving the template here. Then this part is transferred to the other end of the viral RNA here and extended further. At the same time, the RNA strand is degraded, except for this small part, which then works as a primer for another polymerization, which is then moved along. It's pretty complex, you don't need to pay attention to the details. RNase H activity is important in several locations. This primer needs to be cleaved off, and also this polypyridine primer here, which is defined by these cleavages, it's known that it's cleaved very precisely here. So we looked in to see if we could predict these known cleavages of the RNase H. So this is this polypyridine tract, so it's here, and we actually do predict this to be cleaved quite efficiently just next to this known primer sequence. Yes. Then it's known that this primer, tRNA primer, is cleaved off quite efficiently, and it's interesting that this particular tRNA is used by more or less all lentiviruses as the primer, and there are some suggestions to why this is so conserved. We again predict very efficient cleavage at precisely the right location using our model for HIV RNase H cleavage. And then when we look at the entire genome sequence, we observe that the PDS sequence, the primer binding sequence, which is the reverse complement of the tRNA, actually is also very efficiently cleaved, which was not described in the literature. So when we look at all tRNAs in the cell, then this list 3 tRNA is actually the one that is cleaved most efficiently, both in the forward and the reverse orientation. So we think that this may be contributing to this very conserved usage of list 3 as a primer. It would be really hard for the virus to change the primer and having these that the sequence needs to be cleaved efficiently by RNaseH. I have to mention that there are also other reasons why this could be so shown that there are some interactions with other parts of the viral genome, and also there seems to be some selective packaging of this particular tRNA sequence into the viral particles. Finally, then we looked across this entire prediction of cleavage sites and looked on the district, or how well spaced they were. So here we are looking at the red points here is what we observe. So we observe a certain count of cleavages that are separated by 13 to 19 positions, and so forth for these other positions. Then we tried to shuffle the genome in many different ways, seeing what would be expected by chance, what would the distribution of cleavage size be, and for this particular size range, we see that it's very unlikely. There are many more efficient sites spaced by 13 to 19 bases than what one would expect. So you can imagine that when the RNA is reversed, transcribed by the HIV, it's important that you have these periodic cleavages of the template in order to have the RNA released from the newly synthesized DNA. And this is what we expect is going on here. Yes, I think we can use this sequencing-based assay to read out preferences of this enzyme, and it could potentially be used for other nucleases. So we can see that the bacterial and the human RNase H has nearly identical sequence preferences, and we can show that this correlates with the effect of gapnets. Then the HIV RNase H has some quite distinct sequence preferences, and it may help explain this universal use of the tRNase 3 as a primer, and perhaps also shows that the genome sequence has been adapted for efficient cleavages during polymerization. That's what I have said here. So, very brief summary. We looked at off targets of gapnets using this LibSec strategy. We used the H-bar sequencing strategy to look at the sequence preferences of RNase H, and then I think that it seems that RNase H has these specific sequence preferences, and this is actually something that Roche is now using when they design their oligos. So this is another filter that they are putting on their oligo predictions. So this has been collaboration between my group and Roche, especially Morten Lindahl has been a very good collaborator. So Jakob did the LibSec study, and Lukas did the H-bar study, and this first study was financed by the Danish Council for Strategic Research, part of this collaboration. And then the second one was actually funded by Roche. They have an internal postdoc program that funds collaborations between the different Roche departments and academic groups. Lukas did his PhD in my group and then moved out there, so we kind of continued some of the things that he would do while he was in my group. So I think that's it. So we have to answer any questions if you may have them.
I'm from social science.
Sorry about that.
No worries. So let me just state a little bit what I got from it, and then I actually have two questions. The first is, okay, an oligo is a short sort of sequence from the DNA. And then you go on cutting this in all kinds of ways. And you want these oligos to perform some tasks for you ultimately that are more or less useful. Maybe this is wrong already.
No, I think it's
okay.
pretty
So I have two questions. One goes to the kind of task you want them to do that is broad significance.
Yes.
I assume it's something like figuring out what kind of, by the math, what kind of properties the stuff you match them to have. That means diagnostics, perhaps, and perhaps ultimately cure. That's one question. But I also have a question that relates basically to the efficiency of your search. When you search to cut them in all kinds of different ways, do you, for example, use machine learning or other tools like that? So these are the two questions.
Okay, so I think if I knew that there would be people for social science, I should have included more introduction, I think.
No worries.
Yeah, so the ultimate goal is to improve these oligos, and the reason for making these oligos is to make them as drugs. So you want them to interact with a specific gene sequence and very specifically downregulate that gene, but not all the other genes that are present in cell. So that's the entire idea of it, you can say. So I think that RNase H is important for other reasons as well, but the very applied and the reason we got money to do this is to make these small oligos, so there's a small piece of DNA basically, make them efficiently downregulate a specific gene, but not target all the other ones. So that's the starting point.
And then there was the other question regarding, we also do kind of search algorithms and so on and so forth. So it's just, do you also use advanced machine learning tools to search, or how do you do that?
Not really machine learning, I would say, but the models that we build from the data are quite efficient. I would say this is based on these dinucleotide models. So taking in.
So you have enough constraints basically to avoid blind search.
Yeah. So we come. Yeah. I didn't have time to go into complete details with this. So here, for instance, we use our data to train a model that can predict the efficiency of cleavage. And then, of course, it works very nicely on the data that we trained it on. But here we're then using that we actually have two different experiments. We had different designs at the very beginning here. We have some different designs here. So we exploit that we can then train the model on the one data set and test it on the other.
Right.
Yeah. To make sure that we are not just re-showing what you would expect from the same data set. I wouldn't call it real machine learning, but it's a pretty advanced model.
I think I sort of get what you mean. In that sense.
Can I follow up on your first question?
Yeah.
Because I was also interested in knowing the outcome of this. So how can you avoid that you don't have like off-organ target specific side effects of these drugs if you use them? So some proteins are important, let's say in the brain, and also in the periphery. You have a diseased brain, but not a diseased periphery. How do you avoid that you get side effects?
I mean, of course you could target it to a gene that is only expressed in your target tissue, or quite specifically. In that way, it doesn't matter if the oligo goes into some other tissues, because there will be no target. So that, of course, is one possibility. then I know that all these companies are trying to ligate these conjugates to the oligos to increase the uptake into specific tissues. And of course, if you could, so it seems that this GalNAc is very efficient at routing the oligos to the liver, so we get really high concentration in the liver. And if you could find other conjugates that could target it to other tissues, that would most likely improve the specificity. But of course, it is like it's given as a systemic treatment, so you could risk genes being downregulated in other tissues as well.
I'm a theoretical physicist. I appreciate what you say, but so what makes your lab unique? I mean, I assume there are many people doing the same, right? So in which sense yours is cutting edge?
This is a tough question, I think.
I
I think that using sequencing for this type of thing is actually only a couple of groups have done this. I think, especially the last one here, I think there's only one other study that has done something that is somewhat similar to it. So they looked into an RNase P, which is another nuclease, but it's working on a single stranded.
So if you had to, following up on the question, if you had to mention other groups that with even other type of techniques that can outbeat you, which one would it be?
They should beat us. I don't think anyone would do it on RNase H now.
But because they don't think it's interesting, or? I mean, they can be anything. Sorry, my English. They might say that's actually relevant, right?
Yeah, so they may come up with some idea that I don't have to do it a better way than what we did. That is possible. But I think there's only one other nuclease for which the preference has been mapped in the same way. Of course, you have many nucleases that have very specific sequence preference, so you know perhaps restriction enzymes, which cleave DNA, double-stranded DNA at a very specific site, which is used for cloning and so on. So for this type of thing is not really necessary. So it's because these enzymes has more subtle preferences that it's worth doing.
So I guess that question.
Yeah, I have two questions. I like that a lot. So first of all, I wasn't really clear to me. So you mentioned it has been studied for a number of years, this technology to actually target specific genes. What has been the sort of the limitation? Is that the efficiency or the specificity? That was the first question. The second question is, now you have the preferred sequence. Were you actually able, or maybe I missed it, so were you actually able to go back into your first study to see if these off-targets, these substitutions actually were then closer to the preferred sequence?
Yeah. So I think that many of these drugs has failed because of toxicity, it seems. So and it's a little bit unclear why. So of course it's. I think many. I'm not really in the business of making these oligos. It's more that I'm collaborating with them. But of course, there's a bit of somewhat concern that why didn't we, working 15 years, why are there not more drugs coming out of it? So it's a bit unclear what this toxicity comes. It may be related to these off targets. I mean, we put in the oligo and it's clearly downregulated the target, for sure. But it also has some of these off targets, and you can also look at this with other RNA sequencing and so on. You can see that other genes are regulated as well. So, I mean, one hope could be that things like this could improve the molecules that are tested so that fewer off targets are there. But there may also be other ways of toxicity, oligos binding to proteins, doing other things that cause these things to happen. The second question was
So if you saw an enrichment in sort of your off targets, if they were closer to the preferred sequence than the what's that pattern?
We haven't looked. Yeah, perhaps we should look. Because in the first study, we are kind of limited. We only have these the few oligos we used. So we have the target is varying, you could say, but it's the same oligos all the time. So we have very limited data to look at. But perhaps it could be interesting to see if we could tease something out of it. I would like to redo the first study again, not with this total degenerate sequence, but kind of using array synthesis or something like that to make it a more focused library that tested specific hypotheses. So we have a false in position 3 and then test this with different sequences. So if we could do this kind of test if we decided on the sequences in the libraries in advance rather than just taking this total degenerate. It's hard to get money for doing follow-ups.
Last question. So it seemed pretty clear from the cross-section of the mouse that had been dosed with the labelled oligo that it is not going across the blood-brain barrier at all, because the brain was just like white space. But you can think of situations where this could be pretty valuable, for example, to treat amylopathies in the brain. So if you wanted to get one of these across the blood-brain barrier to treat something like that, what strategy would you take?
I mean, there are actually development programs going for brain targets, and then you inject in the spinal fluid. And
Right,
this,
so you would just go straight for the...
yeah. You could perhaps also go directly, but this actually seems to work fairly efficiently, and you get pharmaceutical action in the brain. So I know that there are programs, both Ionis and Roche has programs that are focused on brain targets. But of course, I mean, for liver-based therapy, then it could be an advantage that is not going to the brain, as you say.
Okay, thank you very much, Jeppe. Thank you everybody for coming. If you have more questions, I guess you can approach Jeppe.